All right, welcome to our next video. This is a fun topic because now we get to combine our new knowledge of amino acids with our background knowledge of weak acids and bases and really start to think about the charges on these various molecules. So we're going to apply the concept of weak acid-base equilibria to predict those charges, and we're going to talk about something called the isoelectric point, or the pI of the amino acids. So let's just think about pH in general. If you have a beaker of water, you know that pure water with nothing else in it would have a pH of 7, but that doesn't mean it's all just water. We know that there was some um, autoionization, and so even in what we think of as pure water, we've got some hydronium ion, we've got some uh, hydroxide ion. We know pH is a measure of that hydronium ion, right? We remember pH is negative log of that concentration. And it turns out pH is incredibly important to living systems, and in particular, controlling that pH is crucial for biological systems. So living systems generally are buffered to control the pH. So there'll be some other weak acid base in here that will help us control that pH. So it'll have some of the acidic form, the weak acid form, and some of the weak base form, that A minus, protonated and deprotonated form. Let's think about what happens when we put an amino acid in that water. So if we drop an amino acid in the water, it has two functional groups that can be either protonated or deprotonated, maybe a third, but alanine just has methyl for its R group. So we'll wait and talk about those. So to know if these will be in their charged state or uncharged state, we need to know the pKa's. We can look those up. Now they'll differ a little bit based on various conditions, but the pKa here of our amino group is about 10, and the pKa here of our carboxyl group is about 2. And if I go back and I think about my Henderson-Hasselbalch equation, pH equals pKa plus log of the deprotonated over the protonated version, I can think about each of these functional groups separately. So let's think about what happens if I drop my alanine into a solution that has pH of 7. Let's think about just the amino terminal. So pKa would be 10. When I look at this, I need this whole piece to give me a negative number, in particular, -3, right? To make my math work. And if I need that to give me a negative number, I know that this ratio must be less than 1. I must have more of the protonated version. So I can look at this and say, you know what? I'm going to expect this to be in the NH3 plus version. I'm going to expect it to be protonated. All right, we're going to do the same for the other one. Now I still have my pH of 7, but my pKa is 2. Now, mathematically, I can look at this and say, you know what? I need this ratio to give me a positive value. And I know that if that's the case, I need this ratio to be more than 1. So that carboxylate group is going to be deprotonated. We're going to have more, a lot more, of the deprotonated or the base version of the molecule. And this is what we find for amino acids at pH, physiological pH, and that's why we oftentimes write them this way instead of NH2 and COH. All right, so now what we want to talk about is, well, first of all, let's define this, although I think I've written it somewhere else in these slides, but this would be Zwitter ionic form. It's a fun word, right? Zwitter ionic form is the form where our amino acid is overall neutral. So that would be this case because we've got a +1, a -1, so we get overall neutral. All right, let's take a look. Couple more terms. Okay, so ampholyte, something that has both acidic and basic pKa values, and this is most biologically relevant molecules. So when we look at our amino acid glycine here, whoops, we can see that we've got two areas that can be charged, right? We've got our amino terminal and our carboxylate terminal. At really low pH, we'd expect both of them to be protonated in their acid form. And in this case, then that gives us a net +1 charge. As we start increasing the pH, the one with the lower pKa will deprotonate when we pass its pKa point. All right, and so now we're in our Zwitter ionic form, we're neutral. And if we keep ramping up that pH, we'll eventually deprotonate the amino group, and now we have a -1 charge. So notice with each pKa we pass, essentially, we lower the the net charge by 1. So we always start at a higher charge at low pHs, and then it gets lower. All right, this chart may look familiar from when we did polyprotic acids. This is very similar. And here's what we're looking at, right? So we have an amino acid right here in the pink box that corresponds to this pink line. So at low pHs, I would expect that both the amino group and the carboxylate group are protonated. Okay, and so that's what we have almost all of here. So here's our concentration. If you remember back to when we did polyprotic acids, we mapped this would be a pKa value, this would be a pKa value. We know pH equals pKa plus log of A minus over HA. So we know at those pKa values, when pH equals pKa, this has to be 0, which means that ratio is 1 to 1. We have equal amounts. So what we see is as we start increasing our pH, we start deprotonating. This would be the first, the carboxylic acid, the acidic group. So some of those start losing their protons. Well, that means that we start having this molecule, so its concentration starts going up. At the pKa, they meet. We've got half and half. We keep ramping up that pH, we really drop down that concentration of the one that had the acidic version. And now we really have a lot of that Zwitter ionic form, the one that has a net neutral charge. But if we keep going, we're going to start getting near the pKa for the other functional group here. And as we start getting closer and closer, this one starts donating some of its protons until we get to the net equal spot, right? Where we have equal amounts of the A minus and HA. It keeps going down and we start building up this one where both groups are deprotonated. Okay, that's kind of what that looks like in terms of a graph. We want to we define Zwitter ionic form. The isoelectric point, or the PI, is the pH at which the average charge, okay, so the pH at which the average charge on the molecule is 0. So if I look up here, this is the molecule I'm talking about, okay? And so I want to know what pH that is. Well, if I look at my graph, it's going to be right between those two pKa's. Okay, if I am at a higher pH, if I'm up here, well, I'm still pretty close. A lot of them are neutral, but you know what? I'm going to start having more and more of this. So it's tilting now towards the negative. And if I'm below the PI, if my pH is below the PI, well, you know what? I still got a lot of this, but I'm getting more and more of this. So it's tilting towards the +1 charge. And right here is where we would expect it to be neutral. So we can estimate that by averaging the two pK values. And we're going to do that on the next slide here. So when you look here, I would like you to try these two. So you want to calculate the PI for each of these amino acids. All right, pause and see if you can figure that out. All right, if I look at glycine, you can think about kind of a number line here. I've got two possible things. I've got my amino terminal and my carboxylic acid terminal. And I know that my amino terminal can be NH3 plus or NH2. You would normally write all this out. Carboxylic acid terminal can be COH or COO minus. So if I think about those, let me redraw this a little higher. I know that my switchover points are going to be those pKa's. So I've got glycine, so I know pK1 is 2.34. And I know pK2 is 9.6. pK1, the lower one, will always be the carboxylic acid side. The pK2 will always be the amino terminal. So when I look at this, below this first one, I expect both to be protonated. So below that, I expect to have NH3 plus and COH. So I can see that I'm going to have a +1 charge. All right, let's highlight that. But once I pass that 2.34, I'm going to expect that I've deprotonated this one. So in that middle section, I've still got NH3 protonated, but I've deprotonated the CO minus. So now I have a net charge of 0. Okay, here's the section that I'm going to average out to get my Switter ionic form. And then if I keep going up, right now, find another color here. I'm going to also deprotonate this as I pass that next pKa. So now I've got those two, and that would have a -1 charge. So you can see it drops down by 1 each time. But to calculate the PI, isoelectric point, you're going to calculate the two, or you're going to average the two pKs on either side of the Switter ionic form, and there we would have it. All right. For one that does not have an ionizable side chain, you've only got two pKs. So we could look at isoleucine, and I know it's going to be 2.36 plus 9.68 divided by 2. All right, let's look at one with an ionizable side chain. Much more fun, right? A challenge. So now if I look at this, I have 3 different pKa's. This is a little interesting, a little different from our weak acids, because remember I said that pK1 is always the carboxylate terminal. pKa2 is always the amino terminal. pK3 will be the side chain, whether or not it has a higher or lower value. That'll be pK3. So if I think about my options here, right? I'm going to draw my number line. I'm going to put in my 3 values for aspartic acid. 2.09, 3.86, 9.82. And when I think about this, below the lowest one, they will all be protonated. So I would have NH3 plus COOH, and for my side chain, COOH, I've got a +1 charge, right? This one would be the side chain one. This would be our terminal one, or carboxylate terminal. So as I pass 2.09, hey, this one's going to deprotonate. So now I've got NH3 plus COO minus COOH. Look at that. There's my Switterionic form. I've got a charge of 0. We could keep going. If we wanted to know what the charge would be between 3.86 and 9.82, as I pass that 3.86, I am still going to have the amino terminal protonated, but now I've deprotonated the side chain. So now I've got -1 and -2. So if I want to calculate my PI, I look for where it's 0 and I average those. 2.09 plus 3.86 divided by 2, 2.98. So above that, we start tilting. So right here, right, is our PI. Above that, we start tilting more towards the negative. Below that, we start tilting more towards the positive. All right. This is just pictures of what we just worked out mathematically. So if you want to kind of follow along going from here to these pictures, you can certainly do that. Let's take a look at a few practice questions. Amino acid histidine, we said it was unique because that side chain had a pKa of 6. And for that reason, it's very commonly in active sites of enzymes because it can act as an acid or a base. And we want to know the ratio of protonated to deprotonated form of the side chain, histidine pH 8. I suggest with all of these questions as we go through the videos, you should already be able to do this if you think about it. So pause, try it on your own, and then unpause and see if you agree with my math. All right, I'm going to start with Henderson Hasselbalch. And I know that the pKa is 6, the pH is 8. So I know I need this log value to essentially give me a value of 2, right? Positive. So first of all, I know that this ratio has to be a number greater than 1. And what we'll find, since we're taking the log, you could work it out mathematically, or you can think it through just logically, and we'll find that, and did I do that right? Yep. We want the ratio of protonated to deprotonated. We are above our pH is above the pKa, so we should have more deprotonated. And so we'll find we have a 1 to 100 ratio. All right. And you remember, you're welcome anytime you want. Come by, ask questions in our drop-in hours. All right. Take a minute and try this. This is not the question I thought I was going to. Here we go. We want to draw the structure for the amino acid alanine with the correct charges pH 7, pH 2, pH 12. To do this, you'd have to look up your pKa values. Your pKa value for the amino terminal is, what are we on? Alanine is 9.69. And for the carboxylic acid side is 2.34. All right, pause, see if you can draw these structures. All right, I'm going to start at pH 2. At pH 2, I'm going to expect them both to be protonated because I'm below the pKa's. All right, next, if I look at pH of 7, now I am above this one, so I'm going to expect it to deprotonate. And if I look at pH of 12, now I'm above both, so I expect them both to be deprotonated. Okay, so I could ask you to calculate ratios like we did here. I could ask you to calculate the PI value like we did here. I could ask you to draw structures and tell me what the structures look like at a specific pH. Let's take a look at this next question. At what pH would alanine be an effective buffer? Its pKa are right here, right? This would be the 2.3, this would be the 9.7. All right, think about it, pick an answer. Remember, for a buffer, you have to have a significant amount or decent ratio of both the acid and its conjugate base. pH equals pKa plus log A minus over HA. So we have to be relatively close, usually within about a pH unit of the pKa for this to work. And this acid and conjugate, the acid and conjugate base of the same thing, right? So it's either COOH and COO minus, or it's NH3 plus and NH2. We have some of each. So we could have an effective buffer using that amino acid if it was near either of its pKa's. Not right in the middle. We won't have a lot of either of these two mixes then. Okay, but near either of those. All right, another question for you to pause so you can try it. What is the net charge on isoleucine at pH of 7.2? All right, so when you were going to do this, you would have to go look up your pKa's. The isoleucine doesn't have a charged side chain, and we know that most of the pKa's, and you'll learn this as you're going, for the carboxylic acid terminal are around 2. Like they're 2, let's look at them. 2.3, 2.2. So let's just say, I'm going to guess it's around 2.3. And most of the amino terminals, if you just need an estimate, are around 9 or 10. Okay, so let's say around 10. Now, if I'd ask you to tell me the net charge at pH of 9.7, you're going to want to go look up the specific numbers, but this is really far from both of those. Okay, so we can make a guess. So around 2, around 10. So when I think about this, okay, below 2, they're both protonated. So I've got +1. I know it's going to go down as I deprotonate each sequentially. And so in this middle spot, I would expect to have pH of 0. All right, we've done a lot of practice. Let's talk just a little bit now about the bigger picture. We're going to talk about protein structure in the next video. So right here, we've got kind of a big protein, okay, shown here. We've got pH increasing, so we've dropped our protein in some sort of solution at different pHs. And we're looking at kind of the charge on the surface. And so at a lower pH, those side chains on those various amino acids will be protonated, so we'll have a more positive charge. In this particular protein, it's +6. And as we increase, we'll start deprotonating and our charge will get more negative. These charges would be different for different proteins. But we want to look at how we would do this for a polypeptide. Same exact concept that we did earlier. If we wanted to figure out the charge on a polypeptide at a specific pH, we're going to need to know our various pKa's. So when you look at this, you're going to want to go through here and identify these, all right? And look for our linkages. So if I'm going to actually come in and just kind of, so to the left of that is one amino acid. So here's our amide linkage that links them. So we would have our N-terminal, our alpha carbon, our carbonyl left over from our carboxylic acid, and then it linked to the next one. So there would have been our next amino acid, there would be our next one, there would be our next one. So this one had 5 amino acids. We can look at the side chains to identify them. So this first one would be tyrosine. That second one has 2 hydrogens. That's glycine. Third one's the same. The fourth one, we've got our aromatic ring down here. This is phenylalanine. And then that last one is isoleucine. Now, what's interesting with the polypeptide chain is that the carboxylic acid groups and the amino groups that have been used to bond to make that amide linkage are no longer available to be protonated or deprotonated. Okay, so we don't have to worry about any of these. None of these are going to or be a source of charge. What we do still have to think about are our terminals. Okay, so our amino terminal, which we're always going to write at the left, we're always going to write amino to carboxylic acid. Okay, and we have to think about our side chains. Well, when I look at this, lysine is not going to form a charge. Phenylalanine doesn't form a charge. Isoleucine doesn't form a charge. The only one that could be protonated or deprotonated is the tyrosine. And if you wanted to check that, you can come down and look at your pKa table and say, oh, look at that. Tyrosine does have a third pKa versus, say, isoleucine does not. So then what I've got to figure out is for each of the ones, let's highlight the other one in purple here. Each of the things that can be protonated or deprotonated, which form are they in at pH 4.5? I'm going to say, wow, NH3, I could come down and check, but I know I am below the pKa, so it'll be protonated. So that'll have a +1 charge. The hydroxyl group here, I'm going to go down tyrosine. I'm going to say, wow, that's 10.07. I am below that, so it'll be protonated. So it has a charge of 0 when it has the proton. And then I'm going to come over here and I'm going to say pH 4.5. I'm above the pK, so this will be deprotonated. So my net charge is actually 0 in this case. All right, that is it. In the next video, we'll start really looking at protein structure.